Okay, so good afternoon and a very warm welcome to this innovation workshop, part of the On Helix 2022 conference. And this workshop is hosted by Catalent. And the subject of their talk is going to be their GPEx Lightning cell line expression technology. So just a few words from me before we kick off. So I'm delighted to be opening this session hosted by Catalent, who've been a One Nucleus partner for a number of years now. So a global leading provider of advanced delivery services and manufacturing solutions for your drug discovery and development projects. So I'm going to shortly hand over to Sayali, and she's going to introduce the session and guide us into the presentation. Please do use the chat box for your comments and questions on the content, and there'll be time at the end to discuss the topic and any of your questions, or even if you are thinking of utilizing a similar technology, I'm sure the Catalent experts can be on call to help you today with your questions. So we're also recording the session, so you can catch up post this afternoon if you'd like to revisit some of the content. And yeah, with that, I will hand over to Sayali to guide us into the presentation. Thank you very much.
Thank you, Alicia. So my name is Sayali Benkale, and I'm a marketing communications specialist at Catalent, and I'll be your host today for this webinar called Leveraging Proven Technology to Speed Path in First-in-Human Studies with GPEx Lightning, co-organized with One Nucleus and presented by Gregory Black, Vice President of Research and Development, Biologics at Catalent. Before we begin, I'll address a few items. On the bottom of your dashboard, you notice there's a Q&A chat box where you could also find resource links for you to review. Please feel free to access this information at any time during the presentation and after, and I'll post those links once we've started the session. And again, as Alicia mentioned, please feel free to submit your questions at any time in the presentation, and we'll be sure to answer them at the end of the session in the live Q&A. And if we run out of time, we will follow up with individual participants after the webinar. With that being said, I'm delighted to introduce the speaker for today's session, Gregory Black. Dr. Black is the Vice President of Research and Development at Catalent Biologics, with overall responsibility for development and acquisition of new products and technology. He has published over 60 research papers and has authored over 3 book chapters, and his work at the University of Illinois was recently highlighted in a book about the greatest advances in the first 150 years of innovation at the university. Over to you, Greg.
Thanks for attending today. I'm going to be talking about a new technology we developed, we call GPEx Lightning, which greatly shortens timelines for development of therapeutic proteins in mammalian cells, and greatly increases the titers that we're able to get from those cell lines in those short development timelines. So the GPEx technology and the GPEx suite of technologies has been around for a number of years. We've worked on close to 1,000 different molecules in different categories. Some of these products are commercial, and numerous products are still in various phases of clinical development. The technology works very well to create highly stable, high expressing cell lines. The basis and sort of the advantage of all of these technologies is the flexibility they allow, as well as the high yields you see from these cell lines. We tend to see higher yields from all the products that we work on. This is due to the way the genes insert in active regions of the cell genome. That allows for high levels of expression from cell pools, stable cell pools, as well as from stable cell lines. We don't use any antibiotic selection or traditional gene amplification as part of the process. And so this can shorten timelines, and obviously you don't need to add some sort of inhibitor or antibiotic to get cell line selection or as part of the cell line development process. We can do complex molecules very easily with the system. So 3-chain, 4-chain bispecific molecules, virus-like particle molecules can be done. It's easy for us to titrate in different genes to get that stable cell pool and resulting clonal cell lines to express all the proteins of interest in the correct ratios. And finally, one of the main advantages of the technology is that the cell lines that result are extremely genetically stable. Each copy of the gene inserts at a unique genetic locus, so we don't have head-to-tail arrays inserting at single sites in the genome. And as the cells go through the normal mitosis process and cell division, that those genes are extremely stable and continue to express throughout the course of the stability study or the course of the scale up and bioreactor production run. The original GPEx technology that's now been around close to 20 years uses replication incompetent retrovectors to insert genes into any mammalian cell line. So predominantly we use CHO cells, Chinese hamster ovary cells, but we have done a number of programs in human cells should that molecule need to be made in human cell lines, and can be easily, the technology can be easily switched to CHO cells as well as any other mammalian cell line. Again, we don't use any antibiotic selection, no traditional gene amplification. The base technology, we get cell lines up to 7 grams per liter. And as I mentioned, numerous commercial products are being made using the technology, as well as numerous products in clinical trials. One of the main advantages of the technology is shown here. This was work done by a large pharma collaborator a few years ago now, where they compared their traditional method of making cell lines to the GPEx process. And so we created a cell pool using GPEx, and they created a cell pool using their traditional technology, which was more of a amplification type process. And then they compared those 2 cell pools, and they selected in this initial evaluation 360 clones at random. You can see all the GPEx clones express at relatively high levels, whereas a lot of the clones from their stable cell pool didn't express or expressed at relatively low levels. And so part of the GPEx process, it's a very efficient way of getting genes into cells, which results in virtually all the cells in that stable cell pool, as shown here, expressing protein at relatively high levels. That's sort of outlined in this schematic, where you can see a more traditional standard cell line development technology, where you have a lot of clones that don't express or express at low level, where you need to look really hard to find those high expressing clones. Whereas with the GPEx technology, all the cells within that stable cell pool express protein, and they express protein around a mean that is a sort of a bell-shaped curve. And so within the cell pool, we don't have to look extremely hard to find those really good expressing, high expressing clones. as part of the clonal cell line development process. About a few years ago now, we took the base GPEx technology and then combined that with a traditional GS knockout cell line approach. And when we combined the glutamine synthase knockout cell line with traditional GPEx, we sort of saw a synergy between the way we were practicing the two technologies, where we saw sort of a 1 + 1 = 3 effect. The way we did the selection is slightly different than traditional GS selection, and the way the expression was designed. And we saw a significant boost in the cell pools, where the cell pools, we actually saw a competitive advantage to those cell lines that had more copies of the GS gene in, and more copies of subsequently more copies of the transgene. And so we saw even greater shift to the right, as I'll show you in a couple slides, of that bell-shaped curve, where the cell pools are expressing at anywhere from 2 to 7 times as much as we saw with the previous GPEx cell pools. That sort of selection that's occurring in the unique way that we're doing that glutamine selections highlighted in these two cell pool examples, where we created 2 cell pools and kept glutamine present in the cell line and grew those cells. Those 2 cell pools are sort of resemble what normal GPEx would give us. And then when we removed glutamine and took advantage of that glutamine selection pressure, we saw a dramatic shift in the number of copies, or the average number of copies of the gene in that cell line, where for the first example, we went from 30 to 60 copies of the gene in the pool. And the next example went from 60 to over 90 copies of the gene in the cell pool. So even though all those cells contained the GS gene, the preference is for the ones that contained more copies of the gene and then more copies of the transgene survive better, and we're able to outcompete the lower expressing cell lines, resulting in a very nice high expressing cell pool. That's shown in this schematic again, where when you compare it to traditional GPEx, we saw a narrowing of that bell-shaped curve and a shifting to the right, where that pool showed a much higher mean expression level. And again, it's much easier to identify those really high expressing cell clones. So now introducing the new technology, GPEx Lightning. So just sort of wanted to go over again what traditional GPEx was. So we insert genes into the cell line using a retrovector. We get multiple copies of the gene inserted at various different sites throughout the genome, highlighted by those triangles in this schematic. Each of those triangles contain the gene of interest that is expressed. And we see very good stability. Again, no gene amplification required, no antibiotic selection with this cell line. With the GPEx boost technology, everything's the same as the previous GPEx technology, except we're inserting GS in combination, so glutamine synthase gene in combination with our gene of interest. This shortens the timeline slightly by a few weeks, and then gives us that big increase in the expression level of that stable cell pool that we've seen with the GPEx boost technology. The GPEx Lightning technology sort of takes advantage of both the GPEx concepts as well as the GPEx boost concepts in a unique way, where we use GPEx and insert a doc sequence, so a small DNA sequence that doesn't code for any protein, but is a recognition sequence for a recombinase enzyme into the cell line. And so we insert that doc sequence using the retrovector technology into the cell line using the traditional GPEx process. We've then selected a cell line that contains multiple doc sequences and shows great growth characteristics and behavior in bioreactors and behavior through a clonal selection process. And then we take that cell line and introduce a plasmid encoding a recombinase that recognizes that doc sequence in combination with the gene of interest and the glutamine synthase gene. And so that recombinase recognizes those doc sequences and inserts the gene into those doc sequences. And then we use that, what we call the boost concept, that GS selection, to skew that population of cells to the ones that have the most genes inserted and therefore expressing the most glutamine synthase as well as the most gene of interest. This greatly shortens our timelines. We're about a month and a half faster compared to GPEx boost. We've seen a dramatic increase in titers, as I'll show you, and it's a non-viral process. So we only used the virus to create the original doc cell line. Everything after that cell line's produced is generating just using traditional transfection-based technologies. So how we generated that doc cell line is highlighted here. Docs were inserted into the chosen cell line, so we used the chosen GS knockout cell line as our base cell line. So we inserted docs using the GPEx technology. We created a stable pool of cells using the GPEx platform, and then that stable pool was put on the Beacon instrument from Berkeley Lights for clonal selection. Clones were evaluated for growth characteristics on the Beacon and how well they performed on the Beacon instrument, since that's our base method we use for clonal cell line selection. And we're evaluated how they grew in our base production media. And we also analyzed how many docs were in the cell line. So we wanted a good number of docs in the cell line that would be accessible for the recombinase, and selected the cells also based on the number of docs in the cell line. The best clones were selected, and then we evaluated those best clones in how they performed during the GPEx Lightning process. So we did a transfection of those clones with the recombinase, as well as a number of different transgenes, and selected the clone that behaved the best during that process, resulted in the highest expression and the best growth characteristics. And that now has become the base cell line for all GPEx Lightning programs as we move forward. So once we had that base cell line, how we practice the technology is pretty standard. It is pretty similar to most other traditional transfection processes. So we start with that doc cell line. We transfect into that cell line a combination of the recombinase expressing plasmid, as well as the plasmid that encodes the GS gene and the genes of interest. After transfection, we do glutamine selection. So we remove glutamine from the media. No other selections performed as part of the process. So we don't use any MSX selection or added inhibitor selection as part of the process. Just removing glutamine is the only selection pressure that we put on these cells. Cells were expanded, frozen, and then we performed productivity studies in unoptimized fed batch culture conditions using commercially available medias and feeds in shake flasks. And I'll also show you a little bit of data where we did some optimization of those conditions to see if we could improve expression from these pooled cell lines. So the process is very, very quick from transfection through selection through a protein production run. Takes about 35 to 41 days, depending on how the cells come through selection. And after those 35 to 41 days, I'll show you a number of different molecules, but we see from that pool production, we see anywhere from 3 to 9 grams per liter production. We've seen up to 9 grams per liter for traditional antibodies. FC fusions, we've gotten over 4 grams per liter in the stable cell pool, and recombinant proteins over 3 grams per liter using the GPEx Lightning technology. So this is an example of an experiment where we did triplicate transfection, so 3 different individual transfections, all with the same antibody. So we wanted to show the consistency of the process. And then once we had those 3 stable cell pools, we put those in unoptimized fed batch conditions and culture them over a normal production run in those fed batch shake flasks. So you can see very consistent cell growth, cell viability, the number of copies of the gene as reflected in gene copy index. This is an index of the number of copies of the transgene. It's not an actual number of copies of the gene. The higher the index, the more copies of the gene are inserted. But again, doesn't mean that there's 5 copies of the gene inserted into these cells. It's more than that. It's a gene copy index here means it's somewhere in the neighborhood of sort of 20 to 40 copies of the gene inserted. And you can see cell productivity, again, very consistent. For this particular antibody, we're in the 6 to 7 gram per liter range from a stable cell pool before optimization and before clonal selection. Here we wanted to see if we could do a little bit of optimization and push that stable cell pool even higher. So we identified a different set of conditions where this cell line was able to get up to close to 9 grams per liter or a little over 9 grams per liter. And again, similar cell viabilities over the days in culture. Here's another different antibody. Again, the triplicates show very, very consistent behavior. This is a lower expressing antibody. At this stage in the unoptimized condition, somewhere in the 3.5 to 4 gram per liter range. Again, each of the pools exhibited very similar numbers of gene copies in the cell line. We did some optimization here, and we're able to get one of the pools up to, you can see right around 9 grams per liter again. So a pretty dramatic increase from 4 to about 9 grams per liter with a little bit of optimization. Again, this is from a cell pool before any clonal selection. Another antibody done in triplicate, again, very consistent results. This was again around a 3.5 gram per liter production in the stable cell pool. This particular antibody, we just did a single transfection, but again, this was tended to be a higher expressing antibody, so in the neighborhood of about 6 grams per liter for the cell pool, and very similar cell growth and cell viability curves to what we had seen for the other 3 antibodies. This is data from FC fusion, where we again saw similar cell growth and cell viability curves to what we had seen for the antibodies, with the production right around 4 grams per liter for this particular FC fusion and this individual cell pool. One of the things I told you about GPEx and the GPEx suite of technologies early on was that we see very stable cell lines. And with the GPEx boost as well as the GPEx Lightning technology, one of the other things we've observed is we see very stable expression even in the cell pool. So before any clonal selection, these cell pools behave very similar to a clonal cell line in regards to stability. So in this experiment, we used 3 different cell pools, each expressing an FC fusion, the same FC fusion. And we also looked at the stability study done in 2 different media. So ACTI and FB media are 2 different media where we're looking at the stability in those media. And so what we see over the 40 generations, we did this stability study that very consistent titer and very consistent specific productivity, even in a cell pool where you have a diverse set of clones that are part or making up this cell pool. We see a very consistent expression of the pool over that time. And we think this might allow some of our partners to consider maybe taking some of these cell pools, using them for production, GMP, and taking them into clinical trials, or be used as maybe making molecules that are used as part of the GMP production process. So I mentioned the GPEx Lightning greatly shortens timelines to clonal selection, the way we do our clonal selection process here. We use the Beacon instrument, as I mentioned previously, and then we usually go into a Amber bioreactor system, either the Amber 15 or the Amber 250, and screen a number of clones that we selected off the Beacon in various different conditions. Till we get that Amber data from DNA cloning, till we have that Amber data and are ready to scale up to a larger scale bioreactors, is about a 3.5 month timeline with the GPEx Lightning process. So I'm going to show you data from one clonal cell line program we did for that FC fusion. So clonal cell lines were isolated from the Beacon. We chose the top 25 lines off the Beacon. And then we analyzed those top 25 lines in unoptimized fed batch spin tube culture. And for this particular experiment, we chose the top 3 clones strictly based on titer, and moved those clones directly into a 60 generation stability study, where we froze down those clones after certain generations throughout the stability study, and then at the end of the study, analyze those clones and how they behaved in fed batch culture in shake flasks. One of the nice things about the Lightning technology is, even though those high expressing pools are very very consistent, and we narrow the sort of that bell-shaped curve I talked about earlier. There's still a large amount of diversity within that pool, and that's highlighted here, where we see a diversity of expression, as well as a diversity of number of the docs that contain a gene in them. So we have a number of different gene copies of the cell line represented in that stable cell pool that we isolated clones from. So anywhere from, I think, around 20% of the docs were filled up to, in one case, 100% of the docs were filled in one of the cells. And you can see the more the docs were filled, the higher the titer in these unoptimized fed batch conditions. So still a diverse pool that we get to select those best clones from. So those 3 clones we selected, totally based on titer, moved into a stability study. And you can see in this particular example, we did a little over 60 generation stability study. One of the clones expressed around 5 grams per liter. One of the clones expressed about 6 grams per liter. And one of the clones expressed around 7 grams per liter. So without any selection based on stability, just selecting based on titer, we got, as we would expect, 3 extremely stable cell lines from the GPEx Lightning technology, and each of these cell lines capable of moving forward into further development and potentially further optimization. So in concluding, GPEx Lightning results in very good titers in stable cell lines, as well as these stable cell pools. Both the stable cell pools as well as the stable cell lines show remarkable stability in different media, and show that they're very good cells for further development. The Lightning process results in much shorter timelines, and it results in, like I said, those really high expressing pools. So very early on in a development program, basically 35 to 40 days into a development program, we have a pretty good idea of what sort of level of expression we're going to get to with that particular molecule, and any challenges we might come across with expressing that molecule at those high levels. So we're very excited about the technology, and look forward to developing even more data around it, and moving numerous client programs forward using the technology.
That's great. Thank you, Greg. We are now going to open the floor for questions. And as a reminder to our attendees, please feel free to post your questions in the Q&A function. I think we've got...
Yeah, we've got one there. So thank you for your comments, Marcel. So Greg, impressive number of CLD projects. What is cell line development, projects? What is the mechanism behind targeting active genome regions? Is this inherent to retroviral delivery?
Yeah, so it's inherent to the retrovector we use. They tend to preferentially insert in more active or open regions of the cell genome. This has been shown through a number of different publications. So we're using that sort of inherent ability of the vectors to insert, and then accentuating that again with our ability to consistently target those sites with the new technology.
And also interested in why you do not have to prove stability.
Yeah, we still have to prove stability, but typically we take it off the critical path for cell line development, meaning we've never seen a genetically unstable GPEx cell line. So we just wait, and we once we lay down the master cell bank, we just confirm the stability of the cell line at that time point. So we have enough experience now with how the cells are going to behave that we're confident enough that the stability is going to be fine. So we just confirm it at the master cell bank stage. So that's how we perform sort of 95% of our programs, is we move forward with development, select the clonal line strictly based on how it's performing from an expression point of view, from a protein quality point of view, from a cell growth point of view, and then just once the bank is laid down, just confirm that we see good stable expression over 40, 60, 100 generations.
Excellent, thank you very much. Greg, could you elaborate on how GPEx Lightning would be performed for complex molecules such as bi-multispecifics and fusion proteins?
Yeah, so one of the really unique things about the technology and the way that recombinase works is that we can flip genes, basically sort of almost titrate genes into those docks. So the base cell line has about 200 dock sequences, and so we can express two genes at the same ratio to one another, or we can express them at different ratios to one another. And we know that recombinase is going to flip the genes into the stable cell line at whatever ratio we do that original plasmid transfection with. So we've done 4-chain bispecific antibody molecules where we're expressing 4 different proteins, or need to express 4 different proteins. We've seen pooled cell lines expressing 4 proteins at over 4 grams per liter, and clonal cell lines after selection over 10 grams per liter. And when you're doing those 4-chain bispecifics, you need to make sure that the ratios are coming together to form the best quality protein that's possible. And like I said, it's really easy for us to do because we can easily titrate the genes relative to one another. All those dock sites basically allow the expression of the gene at high levels, so that makes the expression sort of directly correlated to number of copies of the gene that end up inserted in that final cell line.
Thank you, Greg. What are the next steps for this suite of technology?
Yeah, so right now the technology works in CHO cells, which the majority of our programs are being performed in. The next step are moving it to other cell lines. So we're looking to move it into couple different human cell lines. We believe the same concepts that we use to develop the current CHO system will also be directly applicable to different cell types. And so we're looking to migrate the technology into those other cell types, and then as well as just improving the functionality, the application of the current technology in CHO cells. Thank
you, Greg. And of course, a lot of what we're aiming to do with the conference is to connect biopharma with those who are providing services such as yourself to support their discovery projects. So leading into that, is the GPEx technology available for out licensing?
Yeah, so the technology is very simple to use. Some of the data I showed is sort of confirms that, where we you know, performed experiments in triplicate, and they basically overlaid one another. So we are entering out licensing agreements where our partners take the technology in-house, practice it in-house, and are seeing similar results to what we're seeing. So that's always good to see. And it, like I said, it's really easily transferable with the base cell line and a few plasmids, and the system can be up and running in labs around the world.
And would there, sort of leading on for that, would there be sort of significant cost implications for biopharmas to consider if they did that, bearing in mind we sort of have a range of different company sizes in our network?
Yeah, and that we have different structures associated with that. So there's research licenses, commercial licenses. What I can say is that, you know, for standard antibodies right now, where we haven't seen, you know, some sort of expression issue, our clonal cell lines are consistently in the 7 to 15 grams per liter range, and so we think that really high titer that you can consistently get with the technology sort of allows partners to pay for any cost that is associated with the technology very quickly.
Thank you, Greg. And then sort of looking to compare to sort of competing cell line technologies, how would you distinguish Catalent's technology here?
Yeah, I think the unique things are, and have always been over all the different iterations of the GPEx technology, are the unique stability of the lines. That's just due to the inherent way the genes insert into the cells, that single copy at multiple sites throughout the genome. And then the simplicity of use and the short timelines. So like I showed, 40 days after transfection, we were already producing in the neighborhood of 3 to 12 grams per liter from a cell pool, where our partners can generate material, a lot of material, very quickly in the development process. And so partners can take that forward to do, you know, detailed formulation analysis, or start to develop the downstream purification process, or if they want to do a preclinical toxicology study with that material, or efficacy study, they have enough material very early in the process to do that. So I think it's the stability of the lines, the speed at which you can get that kind of you know, unique data from that stable cell pool, and then the fact that eventually that clonal cell line is going to be highly productive. That sort of is some of the unique advantages compared to other technologies that are out there. And then the final one is again that we highlighted earlier. There's really no true antibiotic or other sort of forced selection that needs to occur to get that high expression. And so you don't have to co-express a antibiotic resistance gene as part of the process, or treat with MSX or methotrexate to get that amplification boost as part of the process.
Thank you very much. So I'd just like to say thank you, Greg, for your time today on the call and for presenting and for fielding those questions. It's really exciting to see the advances in the technology, and of course, in fitting with the theme of the wider On Helix conference, a brilliant example of one of the enablers that is leading to greater innovations, faster innovations, more efficient innovations, ultimately leading to improved patient benefits in our drug discovery and development timelines. Sayali, did you want to say anything further before I close the session?
No, I think that was great. And I would just also take the opportunity to thank Greg and to our participants, and also to One Nucleus and the entire team who helped us organize this webinar and bring this to life. So thank you so much for being interactive and engaging in this webinar and attending today's session. And as always, you can find more details, or if you have any further questions, you can reach out to us at solutions@catalent.com, and more details can also be found on the website, and also the links that were put in the chat box if you would want to read further on this topic. And yeah, that's all from me. Over to you, Alessia.
Excellent. Thank you both very much. Yes, if you can also use the conference app if you'd like to get in contact with Catalent to discuss any of the content or the technology they presented on, or indeed any other solutions that you may think they can assist with. And I'd just like to remind everyone that we will resume innovation workshops tomorrow, 10 a.m. UK time, with a workshop hosted by Partners And. So with that, I wish everyone a good rest of the afternoon or day, depending on where you're dialing in from. And thank you very much for joining. Take care. Thanks. Bye.
Thank you.